Welcome to the Brains on Brains podcast, brought to you by ECNP, where early career scientists speak with the most interesting experts in applied and translational neuroscience.
Welcome to the ECNP podcast. Today it is my great pleasure to talk to Sabine Hölter Koch. She holds a PhD in biology, is a lecturer in behavioral biology at the Technical University Munich, and leads a research group at the Helmholtz Center in Munich. She also initiated a new ECNP course on neuroscience research. But before we dive into the course, Sabine, let us get to know you and your work. Can you start by telling us a little bit about your academic pathway?
Yeah, thank you very much, Luisa. It's great to meet you this way. My academic path, well, I studied biology in Tübingen in Germany and actually went to the University of Sussex in the UK for an exchange because there was the visiting and exchange program of the German Academic Exchange Service already then, a long time ago. And that's actually where I got into neuropsychopharmacology because I could do a semester project there, which really inspired me to choose that path. And I finished my diploma, it was at the time, back in Tübingen, and then moved on to do a PhD in behavioral pharmacology at the Max Planck Institute of Psychiatry in Munich. And after that, I stayed there actually for a postdoc because that was also the time where I started having a family. And then later on, could move to my current position at the Helmholtz Center in Munich, where I was actually recruited to set up the behavioral lab in the German mouse clinic. And this is where I'm still working.
Yeah, I think Munich is a great place for doing research, but also engaging in academic lecturing, because you also lecture at the university. So you are involved in research, but also in educating the next generations. And this is also what the course will be about. But can you tell us a bit about your research group? What are the central questions your lab is trying to answer?
Well, my research focuses on the translational relevance of genetic mouse models for neurodevelopmental, neurodegenerative, and rare diseases. We're trying to better understand the genetic contributions to different disorders that affect brain function, behavior, and metabolism. And we mainly use genetically altered mice as model systems for that. And together with my colleagues from the German Mouse Clinic and also from the International Mouse Phenotyping Consortium, IMPC, my lab has contributed to the mission of the IMPC to share as a consortium the workload to generate one knockout mouse line for each gene that the mouse shares with the human and phenotype this mouse line comprehensively for all organ systems and physiological functions using 7 males and 7 females, so mice of both sexes. And at the beginning, we decided as a consortium on translationally relevant test assays that produce robust results and harmonize our protocols in experimental cross-laboratory comparisons to ensure that we all measure the same things and that our results are comparable. And then later on, my lab was responsible for the behavioral phenotyping of those mouse lines that were generated and phenotyped at the German Mouse Clinic.
It sounds like a huge group effort, what you're doing there.
Over many years. So this really grew with time, you know.
Yeah, amazing. If you look back over the past, let's say, 5 years, what was the most exciting finding of your group?
Oh, the most exciting one was that we could really use this data. You have to imagine that today the data of more than 9,600 lines is available to actually work with. But we started already, maybe it was 5 years ago, with a smaller set at the time. I think at the time around 4,000 lines had been phenotyped, and we wanted to see if we can already learn something for using a specific phenotype pre-pulse inhibition that is related to neurodevelopmental disorders, to schizophrenia, but not specific to schizophrenia, and actually see, what translates from the mouse to the human. And we used the brain transcriptomic data that's available from the LM Brain Atlas, from the mouse and human LM Brain Atlas, to basically see how schizophrenia genes that we know from GWAS studies and pre-pulse inhibition genes that produce the phenotype in, I think, around 188 lines of these 4,000 lines that were available, and see how these genes come together in disease-relevant brain regions, and if we can actually find a convergence of schizophrenia-related genes and pre-pulse inhibition genes in the functions that are affected. And we found 29 new genes that we're still looking at in more detail if they could give us more insights, for potential new treatments. So that is the best way to make use for us, of this resource. And we're continuing this now with other phenotypes and disease questions.
Yes, I can really imagine that this is so much exciting work, and as you mentioned, also very important insights for next treatment developments, but also to take certain findings into the human model then. And I think we will talk about the topic of the importance of preclinical research in a bit, but I thought we might first dive a little bit into the course. So the new ECNP course is entitled Neuroscience Research, Exploring Models and Methodologies for Translational Neuroscience. How did the idea for this course come about?
That idea was actually already existing when I followed my predecessor, Anton Vespalov, as chair of the ECNP Preclinical Data Forum Network. Anton is very much concerned about the quality of preclinical data, and that was also one of the reasons he co-founded this ECNP network together with Thomas Steckler. But it took actually until 2024 to get together a critical mass of network members to design this course that now covers core principles for robust and translational research, as well as different model systems and biological variables and cutting-edge tools and technologies, I believe, in a very balanced way. So the members of our ECNP network were really essential to develop this course. 10 of the 12 lectures are given by the members of our network. But I must say I'm particularly proud that we could also recruit members of other ECNP networks to give the other 2 lectures on some cutting-edge technologies we felt should definitely be part of the modern course on neuroscience research.
Yeah, this is amazing, especially if you can then recruit colleagues from other networks to engage here as well, and also to translate education on preclinical research to other areas.
Mm-hmm.
Can you tell us a little bit about the format? Will that be pre-recorded lectures? Will there be live sessions as well?
Both. So the course is completely online. It is organized in 12 pre-recorded lectures, which have all a 30-minute duration. And everything is hosted on the ECNP Knowledge Hub. And the lectures are available to watch on demand at the participants' own convenience. All they need is a My ECNP account, but there will also be 6 live question and answer sessions on Zoom on predefined dates. And these Q&A sessions all start in the afternoon, hopefully manageable for most people at 4 p.m. CST. And these Q&A sessions have a 1-hour duration. There are also additional materials prepared, for example, suggested reading resources for each lecture. There are interactive elements such as multiple choice questions and drag and drop activities during and after the lectures. And there will be live assignments to knowledge tests during the lectures. And there's also a discussion forum. So each of the live Q&A sessions will feature the lecturers of the previous 2 on-demand lectures. And these Q&A sessions will include a general Q&A part as well as case-based discussions to allow for the application of what should have been learned during the lectures.
Yeah, this sounds very important because I was wondering, of course, preclinical research involves a lot of lab work and also needs a lot of hands-on training, which is a bit tricky to be delivered in an online format. So how did you try to tackle this problem?
Well, I'm afraid I think the knowledge tests and the case-based discussions is as hands-on as it can get for an online course. So obviously it would be great to, also have lab visits and some practical training formats, but this is not actually part of this kind of online course. But the live assignments and the Q&A discussions and also the discussion forum, I think, offers also opportunities to the different participants to connect. And, I think there are lots of opportunities to maybe engage in practical interactions once you know someone. And hopefully, there will be also things stimulated beyond just the content of the course.
I'm very much convinced that this will happen because people will get to know each other and exchange their skills and their questions. And I can very much imagine that there will be some lab rotations or even some long-term collaborations based on your course.
That would be great. That would be the most favorable outcomes.
Yeah. When we think about, or we have just heard that there are already some participants who have registered, quite a large number, which you are very happy about.
Absolutely.
But of course, there's still much more place, so everyone's welcome to register. But when we think about the target audience, what do you think? Who will benefit the most from this course?
Well, the overall goal is to help clinicians to get a better understanding of the different experimental models, the underlying mechanisms, and also about translational methods that are essential for them to interpret preclinical data and apply that to their work. But we also want to help both preclinical and clinical researchers to engage more effectively in multidisciplinary research and to be able to critically evaluate emerging therapies and in the end to contribute with greater confidence to the development of innovative clinical approaches.
Yeah, I think this is so important because I can say I have never worked in a lab, and perhaps it's good this way. So I've never conducted any preclinical research myself, but especially in the field of brain disorders, it is so important to be able to evaluate findings from preclinical research if you do clinical research, but also vice versa. So in the ECNP Early Career Academy, we, for example, have one interest group who focuses on translation and on the translational exchange between preclinical and clinical researchers.
Great.
So I think, as I mentioned earlier, everyone should feel very welcome to participate in this course.
Absolutely, yes.
The knowledge,
And from every career stage as well,
Yeah, this is also very important to mention that it's not only directed to very early career researchers or only to senior researchers, but everyone at every career stage should feel welcome to join.
Absolutely.
Yeah. To wrap up our discussion about the course, if someone finishes the course, what would you hope for that they understand or feel more confident about?
They should understand the core principles of robust translational research, and they should also understand how the cutting edge tools work, what is their value, but also what is their limitations. And they should feel more confident about engaging in multidisciplinary research. I think that's one of the core aims.
Yeah, that sounds very good. And that you described the course in so much detail helps us also to understand the values of the course. And this naturally brings us to a broader question, because gaining all these skills is not just about doing better science, but also to doing more responsible research. And we have touched a little bit upon the topic that there's an increasing societal but also ethical pressure to reduce animal research. And at the same time, as you mentioned when you described your research, we rely a lot on in vivo models to understand complex brain circuits and disorders of the brain. So to those listeners who are perhaps not involved so much with animal experiments, can you explain what the main issue is at the moment?
Well, that's an incredibly important topic that you raise here, because it affects everybody working with animals, and then it affects our everyday life and work. So there's a huge discussion about promoting non-animal methods or new approach methodologies that is often promoted with the claim that these technologies are there and ready to be used and could immediately replace all in vivo research. Just that the scientists, are old school and don't want to use them, and this is just not true. Um, we embrace, I think the large majority of scientists embraces the development of new approach methodologies, because they also hold a great potential. But we also need to be aware of all the limitations, and we need to do both. We need to develop new technologies. On some of them, we have a lecture in the course. And we still need animal research, both for basic research, but also for certain in vivo assessments that just cannot be modeled with other model systems. And I believe the times are over where we could ignore this kind of debate and let others determine this narrative. We as scientists, young and old, need to be competent in communicating to lay people in a few understandable sentences what we are doing and why that is important.
Yeah, I understand. And what do you think? What are the main barriers? Is it about that regulations become more complex, or is it about how the public perceives animal research?
I think we are really in a battle about public opinion with those people pushing norms. It's fine to push norms, but not at the cost of all the rest. In this battle for the public opinion, I believe we as scientists need to be more outspoken. As I said before, in explaining why that matters what we're doing. We do not need to be able to demonstrate an immediate benefit for society of our own work, but we need to be able to paint the big picture. For example, by explaining that many treatments that we are now taking for granted were initially grounded in basic research in animals, and these experiments at the time were designed to understand biological mechanisms, and it would be great if all of us knew just 3 examples that people can relate to. For example, the in vitro fertilization technologies that were developed in animals and are today used by people with fertility problems, or the development or discovery point inhibitors on the immune cells of animals that led to the development of immune therapies as cancer treatments for humans. I'm sure some people can relate to this, or the development or the discovery of the hepatitis C virus in chimpanzees, which led to the development of an antiviral therapy. And all these 3 examples, for example, got the Nobel Prize. And they weren't maybe seen as what they were worth for at the time that those discoveries were made, but sometime later. And that is probably true for all of our work, although, not every part will directly lead to the Nobel Prize. Every little part helps to increase our understanding of basic biology. And we also learn from negative results, by the way, by understanding what does not work. And anything that leads to really tangible human benefits is the result of all the different components that had to be done before. And that's what we need to be able to explain to the public.
Yes, I fully agree. Thank you so much for giving those three very important examples that also demonstrate, as you mentioned, how sometimes we don't discover, we don't have the big discovery on one day and then everything is clear and understood, but actually it's small steps every day, and sometimes it's a step back, and sometimes it's the negative result that gives us very much information about certain mechanisms or certain methodologies. Yeah. So maybe if we look back to the course, you talked about that animal research struggles a lot with the public image, but then we also talked about that scientists have to be trained not only in the actual lab skills, but also on how to responsibly conduct preclinical research. And what do you think is or if you observe early career scientists at the moment, what is your impression? What are they struggling with?
Well, I would love to give that question back to you, but I can tell you what I observed a week ago when I participated in the ECNP workshop on applied neuroscience in Nice, where where I was fortunate enough to participate as faculty, and there are also career development sessions. And one of the questions that was raised, in the career development sessions, the early career researchers can choose themselves what topics they would like to talk about. And one question was, how to deal with the increasing demands for public engagement and science communication, because almost all of them said that is not part of their curriculum. They didn't learn this in university, but now when they're in grant applications and things like this, they have to fill in forms to basically describe what they're going to do, and they feel that they are not really qualified for this. And this is why we have also one lecture on this in our course. And again, I think all of us need to become really more competent in this and need to train this, and also need to lose the fear of speaking up. And we can train in our neighborhoods, or within our circle of friends. If we can explain in a simple way why what we do matters, to our friends or to our family members, then I think we're on the right track. And then we have to get the right questions back.
Yes. Yeah. So this is basically an invitation to throw a party to your neighbors and explain your research and ask them to ask all the questions.
Start Science Slam more, yeah. Mm-hmm.
Yeah, that's a great idea. But I think for every grant proposal, we have to write those popular science descriptions. And I think many of us are a little bit annoyed by that, but actually this is very important, especially for funding agencies.
I completely understand the annoyance. I remember vividly when I was at that point. It took some time until I understood the value of this. And I think it's really a good training. So we shouldn't underestimate this. And we must not forget, at the end of the day, we are paid by taxpayers' money. And we need to be able, again, to explain to our neighbors, why their taxes, are well invested in what we do. And if we start thinking about this, this way, I find it less difficult to find the right words.
Yeah, but then again, this is also a question of training.
Yeah.
And so it's very important also to train from early career stages on how you communicate your research ideas or your findings to people on different levels
Absolutely.
of knowledge in this topic.
Yes.
When we take a look ahead, because we know that neuroscience and all the methods are evolving very rapidly, and especially when the potential use of AI models comes in, it's a very steep development in the field. Where do you think the field of neuroscience research is heading in the next 10 years?
Well, we definitely need to include all these AI and machine learning technologies. I think there's no doubt about this. Is because this is such an exponential growth here, and we need to embrace this new technology as well as we need to embrace any other helpful technology. But we also, I think, need to be specific what exactly we're talking about. I believe different machine learning approaches to analyze large data sets are really already widely used. But also here, more work needs to be done to make more data sets amenable to these approaches, because this could really hold the potential to pay up on the promise of precision medicine. Yeah, I don't believe that we will get there without incorporating these machine learning and AI tools and the revolution that is taking place there.
Yeah, I see, but I also agree that every model is only as good as the data input is. So
Absolutely. So domain knowledge is still and oversight, is still very important to ensure that these approaches are used in appropriate ways on also quality controlled data sets. Otherwise we risk garbage in, garbage out.
Yeah, absolutely. Yeah, and I also think that because we talk a lot about precision psychiatry or precision medicine and individualized patient care, but to be able to individualize care, we need a complete and holistic understanding about the mechanisms behind different disorders. And there again, we need very solid preclinical data as the first step to understand what is going on or what is not going on. When we look at the time, I think we have had a really insightful discussion, and we have talked about the importance of rigorous neuroscience research and the challenges the field is currently facing, and that one needs to balance scientific progress with ethical responsibility. Before we close, what would be one or two key messages you would like listeners to take away, especially for early career researchers who are considering to enter this field?
Okay, I would like to say we need more scientists who are well trained in good research practice and who understand the value as well as the limitations of a broad spectrum of models and tools. So please do register for our new ECNP course on neuroscience research. We would love to see you there. It covers fundamental principles as well as relevant modern tools and technologies, and it is designed to enable you to engage with more confidence in multidisciplinary research. And we really need you in science. Stay with us.
Thank you so much. I think that's a very important take-home message. And thank you so much again for sharing your insights on your work and how you advance both research but also education. And as Sabine already mentioned, if our listeners are interested in learning more about the ECNP course, Neuroscience Research, you can find all information on the ECNP website and on the ECNP Knowledge Hub. And we thank you a lot for joining us today. Hopefully this episode has inspired you to join the course and then potentially to meet your new collaboration partners at the ECNP Congress, which takes place in Munich in October this year. Thank you so much and have a good day.
Thanks for listening to this podcast from ECNP. The views and opinions expressed in this podcast are those of the interviewee and do not necessarily reflect the official position of ECNP. The content of this podcast should not be used in any way as the basis for treatment decisions.